Hey science fans and curious minds, welcome back to our channel. Today we'll scrabble around into the dynamic world of cell and molecular biology, and our spotlight is on the cytoskeleton and cell motility. Welcome again to this lecture series. This is entitled Moving Bodies, the Cytoskeleton, and Cell Motility. This is Sir Stephen Capilitan from the Biological Sciences Department. We'll start with the overview of the cytoskeleton. And these are the property of the cytoskeleton which remains to be the backbone of our cell. Skeletal system of the eukaryotic cell composed of the three filamentous structures. We have the microtubules, the actin filaments, and the intermediate filaments. In essence, these are the properties of the cytoskeletal components from the microtubules, their intermediate filaments, and actin filaments. And they are categorized based on the subunits incorporated into the polymer, preferential site of incorporation, polarity, enzymatic activity, motor proteins, major group of associated proteins, structure, dimension, distribution, primary functions, and subcellular distribution. And we will be delving into each of these cytoskeletal properties later on. What are the purpose of your cytoskeletal components? They provide structural support and maintain cell shape, positions various organelles of the cell, directs the movement of materials and organelles within the cell, generates forces needed for cellular locomotion, makes up an essential part of the cell division machinery. And this is an example of the epithelial cell, nerve cell, and dividing cell and their cytoskeletal elements. 1 stands for structure and support, 2 stands for intracellular transport, 3 is for contractility and motility, and number 4 is for spatial organization. What is the structure and the composition of your microtubules? It is hollow, relatively rigid and tubular, and they are coming from the protein tubulin. These proteins are arranged in longitudinal arrows called protofilaments. They have 13 protofilaments aligned side by side in a circular pattern within the wall of the tubule, and you can see the image and figures on your right. These protofilaments are assembled from dimeric building blocks consisting of one alpha tubulin and one beta tubulin subunits. These tubulin subunits fit tightly together, so they are like a jigsaw puzzle organized in a linear array along the length of each protofilament. The asymmetric or alpha tubulin at one end and beta tubulin at the other, and also polar, the plus end terminated by a row of beta tubulin subunits, and the minus end terminated by a row of alpha tubulin subunits. There are also we call microtubule associated proteins, and you can see that figure on your right that there's a lot of proteins that are associated from in the microtubule. These are additional proteins called microtubule associated proteins. They increase stability and promote the assembly by linking the tubulin subunits together. An abnormally high level of phosphorylation of one particular MAP called tau or tauopathy has been implicated in Alzheimer's disease. They are also structural supports and organizers. They provide mechanical support by resisting forces that might compress and bend the fiber. Distribution helps determine the shape of that cell. For example, in animal cells, microtubules extend in a radial array outward from near the nucleus, giving the cells a round, flattened shape. They are also agents of intracellular motility, and this determines the shape of the cell. So this is what you see in this figure, that there are protein connecting microtubules and neurofilaments like the plectin. There are motor proteins passing through each of the microtubules, and the microtubules are sliding against each other. So we will explain how it is working in a neuron cell later on. They also traverse the microtubular cytoskeleton. Microtubules serves as tracks for a variety of motor proteins that generate forces required to move the objects within your cell. They can be grouped into 3 broad superfamilies: kinesin and dyneins, which move along microtubules, myosin, which move along actin filaments, They move unidirectionally along their cytoskeletal tract in a stepwise manner from one binding site to the next. They undergo a series of conformational changes that constitute a mechanical cycle. Kinesins, for instance, one motor protein that's the smallest and the best understood microtubular motor, and it is a tetramer of two identical heavy and two identical light chains. As you see the figure on your right, the globular heads bind microtubules and act as ATP hydrolyzing, force generating engines. Each of the head is connected to a neck, a rod-like stalk, and a fan-like tail that binds to a cargo. Each kinesin requires the hydrolysis of one molecule of ATP. Movement is proportional to the ATP concentration, and there's what we call as the hand over hand mechanism. Highly progressive, and it can walk through the microtubule for considerable distances without falling off. As you see the figure on your right, the side view, the top view, and the scale is in 60 nanometer. They have also kinesin mediated organelle transport, where each force generating agents that drive the movement of cargo and organelles, they tend to move the vesicles and organelles in an outward direction toward the cell's plasma membrane. In cytoplasmic dynein, this is a very huge protein with two identical heavy chains, same as the kinesins, a variety of intermediate and light chains, the heavy chain consisting of a large globular force generating head and a microtubular binding stalk. The cytoplasmic dynein moves processively along a microtubule towards the polymer's binding end, opposite of that of most kinesins. As you see the figure on your right, there are heavy chains dimerization domains, and there are tails. We have also motor head, which are composed of AAA plus domains, and stalk and the microtubule binding sites. They position the spindle also in cell division and moves the chromosome during mitosis, positions the centrosome and Golgi complex, and moves the organelles and vesicles and the particles through the cytoplasm. One important thing we know about the microtubule organizing centers is what the centrosomes are. They are nucleation of microtubules that takes place rapidly inside the cell. This is where it occurs in association with a variety of specialized structures called the MTOCs. These MTOCs control the number of microtubules, their polarity, their number of protofilaments, the time and location of your assembly, and the best studied MTOC is your centrosomes. They contain two barrel-shaped centrioles surrounded by electron-dense pericentriolar material, or the PCM. This is a major site of microtubule initiation in animal cells and remain at the center of the cell's microtubule network. In connection to that, the basal bodies and the other MTOCs, a basal body is another type of MTOC that forms at the base of the cilium or the flagellum. Basal bodies are identical in structure to the centrioles, and they can turn into centrioles and vice versa. What about microtubule nucleation? MTOCs share a common factor, gamma tubulin, which is a critical protein in microtubule nucleation. The PCM serves as attachment sites for ring-shaped structures that contain the gamma tubulin, the gamma tubulin ring complexes. This gamma tubulin ring complexes is a helical array of gamma tubulin subunits where the alpha beta tubulin dimers assemble. As you will see, the figure on your right, this is a shot coming from electron microscope, and the artist sketch about your MTOCs and their beta tubulin ring complexes. What are the dynamic properties of your microtubules? They can shorten, lengthen, disassemble, and reassemble. Depending on the cell and location of their microtubules, their stability is determined by your MAPs. These are proteins known as plus tips, which bind into the plus end of the growing microtubules. The enzyme catanin that severs microtubules into shorter pieces. Their disassembly can be initiated by post-transitional modifications called temperature, hydrostatic pressure, elevated calcium concentration, and a lot of chemicals. The microtubules of the cytoskeleton are normally subjected to depolymerization and repolymerization as the requirements of the cell change from one time to another. This is an example of a plant cell, and this is the dynamic character of the microtubular skeleton. And if a typical plant cell is followed from one mitotic division to the next, four distinct arrays of microtubules appear one after another. And what is the underlying basis for microtubule dynamics? During the assembly of tubulin dimers, there is what a GTP molecule, you see that in the figure on your right, which is bound to the beta tubulin subunit. The beta tubulin is a structural protein and a GTPase. A GTP is hydrolyzed to GDP after the dimer is incorporated, and the resulting GDP will remains bound to the assembled polymer. During the disassembly of tubulin dimers, the dimer enters the soluble pool. The GTP is being replaced with another GTP. This nucleotide exchange recharges the dimer, allowing it to serve once again as a building block for polymerization. Then the growth or shrinkage of a microtubule depends on the state of the tubulin dimers at the plus end. The tip, consisting of one open sheet, contains tubulin GTP subunits. The tube begins to close, forcing hydrolysis to the bound GTP. The tube has closed to its end, leaving only tubulin GTP subunits. The GTP tubulin has a different conformation than the GTP tubulin, which makes them less able to fit into a protofilament. The strain from the GDP tubulin at the plus end causes what we call as the catastrophic shrinkage. And there is a term called dynamic stability. This is a phenomena where it describes the microtubule behavior. It explains that the growth and shrinkage of microtubules can coexist in the same region of a cell. A given microtubule can switch back and forth unpredictably between growing and shortening phases. Dynamic instability is an inherent property of the plus end of the microtubule, where subunits are added during growth and lost during shrinkage. There are also microtubules and the cilia and flagella, which we will discuss the structure and function. Cilia, for example, is a hair-like, sometimes motile organelles. They project from the surface of a lot of eukaryotic cells. Motile cilia usually occurs in large numbers on a cell surface with a coordinated beating activity. These cilia move the fluid and particulate material through the tracts, for example, in your respiratory tract. There's usually one non-motile cilia, which is a primary cilia per eukaryotic cell. This cilia acts as a sensory organelle, monitoring mechanical and chemical properties of the extracellular fluid. For flagella, it typically occurs single or in pairs. In a variety of beating patterns, we have the waveforms depending on each of the cell type. You'll see this direction on your diagram in letter B. The flagella is moving at the beating patterns. Cilia and flagella have a membrane that is continuous with the plasma membrane. The axoneme is the core of the cilium. It contains an array of microtubules that run longitudinally through the entire organelle. The axoneme of a motile cilium or flagellum consists of a 9 peripheral doublet microtubules surrounding a central pair of single microtubules known as the 9 plus 2 array. You will see that dynein arms on this figure, which is what we call as your axoneme. Axoneme contains peripheral doublets with A tubule, which is a complete microtubule, and a B tubule, which is an incomplete microtubule. Central tubules are enclosed by a central sheath connected to the A tubules by a set of radial spokes. Doublets are connected to one another by an interdoublet bridge, an inner arm and outer dynein arms that project from the A tubules. You'll see this figure on your right of an axoneme, the central sheath, the outer and inner doublets, the dynein arms, outer dynein arms, the radial spokes, the interdoublet or the Nexene bridges, and what we call as your central microtubules. When it comes to structure, a longitudinal section reveals the continuous nature of the microtubules and the discontinuous nature of other elements. A cilium or flagellum emerges from a basal body. If a cilium or flagellum is sheared from the surface of a living cell, a new organelle is regenerated as an outgrowth of the basal body. What about growth of an intraflagellar transport It is a process through which material is transported along the cilium and flagella. IFT proteins assemble into a protein complex called the IFT particle. This particle line up to form linear arrays called the IFT trains. These IFT particles carry cargo proteins like tubulin out to the tip for assembly. Inhibition of ITP prevents assembly of cilia and flagella. So this is the diagram here is being described in this slide. The dining arms anchored along the A tubule of the lower doublet attached to binding sites on the B tubule of the upper doublet. Dynein undergoes a conformational change, which is a power stroke that causes the lower doublet to slide toward the basal end of the upper doublet. The dynein arms have detached from the B tubule of the upper doublet. The arms have reattached to the upper doublet so that another cycle can begin. Nexin forms an elastic linkage connecting the adjacent doublets, and this limits the extent that adjacent doublets can slide over one another. Sliding on one axoneme side alternates with sliding on the other side so that a part of the cilium or flagellum bends first in one direction and then in the opposite direction. At any given time, these dynein arms on one side of the axoneme are active, while those on the other side are inactive. And these are some extra information about the structure of the cilia and flagella. There is what we call as ciliopathies, a set of seemingly unrelated diseases that all result from primary cilia defects. These include polycystic kidney disease, which are fluid-filled cysts that are formed on the surface of your kidneys as a result of mutations of PKD1 and PKD2 located on the primary cilia. Bardet-Biedl syndrome, or the BBS, is a developmental syndrome caused by abnormal protein trafficking to the primary cilia. Let's move forward with the intermediate filaments. These are strong, flexible, rope-like fibers that provide mechanical strength to the cells, and they are subjected to physical stress. They're a chemically heterogeneous group of structures, and they are divided into 5 major classes based on cell type and other criteria. These are the IF proteins like keratin, acidic, basic, vimentin, desmin, glial, fibrillary acidic protein, peripherin, neurofilament proteins, NFL, NFM, NFH, nestin, lamine proteins A, B, and C. And there are sequence types 1, 2, 3, 4, and 5. And you will see that in the table, their primary distribution in tissues. These are often interconnected to other cytoskeletal filaments by thin, wispy cross bridges consisting of the protein plectin. As you see this photo on your right, each plectin molecule has a binding site for an IF at one end and a binding site for another intermediate filament, microfilament, or microtubule at the other end. When it comes to IF architecture, you will see this photo on your right. Each monomer has a pair of globular terminal domains separated by a long alpha helical region. The pairs of monomers are associated in parallel orientation to form the dimers. The dimers associate in an anti-parallel, just like your DNA, staggered fashion to form tetramers, the basic unit in IF assembly. 8 tetramers associate to form a unit length of the IF. Elongated IFs are formed from end-to-end association of these unit lengths. How do they assembly and disassemble? These assembly steps do not require the direct involvement either of ATP or GTP. The tetrameric building blocks lack polarity, as does the assembled filament, which distinguishes IFs from other cytoskeletal elements. The subunits are not incorporated at the ends of the filament, but into this filament's interior. Unlike other two major cytoskeletal elements, assembly and disassembly of IFs are controlled primarily by subunit phosphorylation and dephosphorylation. We have different types and functions of intermediate filaments. We have keratin-containing IFs. These are the structural proteins of your epithelial cells. They are being tethered to the nuclear envelope in the center of the cell and anchored at the outer edge by desmosomes and hemidesmosomes. They function to organize and maintain cellular architecture and absorb mechanical stress. Neurofilament IFs, on the other hand, is located in the cytoplasm of the neurons in bundles oriented parallel to the axon. So we start with the actin and myosin. In the actin structure, this is the third major type of cytoskeletal element that is involved in intracellular motile processes. An actin filament or the F-actin microfilament is a two-stranded structure with two helical grooves running along its length. Actin filaments can be organized into unordered arrays, highly branched networks, and tightly anchored bundles. All of the monomers within the actin filament are pointed in the same direction, resulting in a polar filament with so-called barbed and pointed ends. This is a major contractile muscle protein and a major protein in every eukaryotic cell. How do your actin filaments assemble and disassemble? Before it is incorporated into a filament, an actin monomer binds to a molecule of ATP. Actin is an ATPase just as tubulin is a GTPase. The ATP associated with the actin monomer is hydrolyzed to ADP at some time after it is incorporated into the end of the growing actin filaments. The initial nucleation event in filament formation occurs slowly in vitro, whereas the subsequent stage of filament elongation occurs more rapidly. Both ends of a filament becomes labeled, but the fast-growing barbed end incorporates the monomers at a rate about 10 times that of the pointed end. In vitro, these preformed actin filaments are added in the presence of ATP. As long as the concentration of ATP actin monomers remain high, subunits are added to the both ends. As monomers are consumed by addition to the ends of the filaments, the concentration of free ATP actin drops until a point is reached where net addition of monomers continues at the barbed end but stops at the pointed end. You can always refer to the figure on your right. Fourth, as filament elongation continues, the free monomer concentration drops such that the monomers continue to be added to the barbed ends of the filaments, but a net loss of subunits occurs at their pointed end. A point is reached where the two reactions at the opposite ends of the filaments are balanced such that both the lengths of the filaments and the concentration of free monomers will remain constant, and we refer this one as treadmilling. These processes can be influenced by a number of different accessory proteins. By controlling this dynamic behavior, the cell recognized its actin cytoskeleton required for dynamic processes such as cell locomotion, cell shape changes, phagocytosis, and cytokinesis. Myosin is the molecular motor of actin. Molecular motors that operate in conjunction with actin filaments. They move toward the barbed end of an actin filament. All myosins share a characteristic motor head domain. The head domain contains two sites, a site that binds an actin filament, a site that binds and hydrolyzes ATP to drive the myosin motor. Whereas the head domains of various myosins are similar, the tail domains are highly divergent. They generally divided into two broad groups, such as your conventional or type 2 or the unconventional. The conventional type 2 myosin. These are the primary motors for muscle contraction. They can be also found in a variety of non-muscle cells. All myosin 2s move toward the barbed end of the actin filament. Among their non-muscle activities, type 2 myosins are required for splitting a cell into two during cell division, generating tension at focal adhesions, cell migration, and turning behavior of growth cones. Myosin 2, as what you see in this figure, consisting of a pair of globular heads that contain the catalytic site of the molecule, a pair of necks, each consisting of a single uninterrupted alpha helix and two associated light chains, a single long rod-shaped tail formed by the intertwining of a long alpha helical sections of the two heavy chains. The fibrous tail portion of myosin 2 plays a structural role so it can form filaments. Myosin 2 molecules assemble so that the ends of the tails point toward the center of the filament and the globular heads point away from the center. As a result, the filament is bipolar, indicating a reversal of polarity at the filament center. Because they are bipolar, the myosin heads at the opposite ends of a myosin filament have the ability to pull actin filaments toward one another. Myosin 2 filaments can assemble transiently when and where they're needed. For unconventional myosins, myosin 5 moves hand over hand fashion similar to that of the kinesin molecule, so at least one of the two heads must be bound to its polarized track at all times. There are several unconventional myosins, including myosin 1, 5, and 6, are associated with various types of cytoplasmic vesicles and organelles. Several unconventional myosins 1, 3, 5, 6, 7, and 15 are localized at various sites with the hair cells of the inner ear. Myosin 6 is distinguished by its movement in a reverse direction, that is, toward the pointed end of an actin filament. Myosin 6 is located at the base of the sclerocilia, where it might connect the cytoskeleton to the overlying plasma membrane. Now let's move ahead with the muscle organization and construction. This is a part of your skeletal-muscular dynamics. The organization of sarcomeres. A skeletal muscle fiber is a multinucleated cell. Each muscle fiber contains hundreds of cylindrical strands called the myofibrils. As you will see this figure on your right, each myofibril consisting of a repeating linear array of contractile units called the sarcomeres. A sarcomere extends from one Z-line to the next Z-line. It has a densely staining M-line that lies in the center of the H-zone. The I-band contains only thin filaments. The H-zone contains only thick filaments. The part of the E-band on either side of the H-zone represents the region of overlap and contains both types of the filaments. Then there's the sliding filament model of muscle contraction. Shortening of the sarcomeres results from the thin and thick filaments sliding over one another. As a sarcomere shortens, the A-band remains constant in length. The H-band and I-bands decrease in width and then disappears altogether. The Z-lines on both ends of the sarcomere move inward until they contract the outer edges of the A-band. Thin filaments of a skeletal muscle contain actin, troponin, and tropomyosin, as what you see this figure on your top. Tropomyosin is an elongated molecule approximately 40 nanometers long that fits securely into the grooves within the thin filament and is associated with 7 actin subunits. Troponin is a globular protein complex composed of 3 subunits spaced about 40 nanometer apart along the thin filament and contract both actin and tropomyosin. Actin filaments of each half sarcomere are aligned with barbed ends linked to the C-line. The thick filament is composed of several hundred myosin 2 molecules together with small amounts of other proteins. The center of the filament is composed of the opposing tail regions of the myosin molecules and devoid of heads. These heads project from each thick filament along the remainder of its length due to the staggered position of the myosin proteins that make up the body of the filament. And the third most abundant protein of your skeletal muscles is your titin. These titin filaments are elastic and they prevent the sarcomere from being pulled apart during muscle stretching. Maintains myosin filaments in their proper position within the center of the sarcomere during every muscle contraction. The nebulin molecules act as the molecular ruler by regulating the number of actin monomers that are allowed to assemble into a thin filament. During contraction, each myosin head extends outward and binds tightly to a thin filament, forming a cross bridge between the two types of filaments. When bound to the actin filament, myosin head undergoes a conformational change that moves the thin actin filament. Since each thin filament is contracted by a team of 100 or so myosin heads that beat out of synchrony with one another, it undergoes a continuous motion during each contractile cycle. Now we move forward with the energetics of filament sliding. Energy is provided by ATPase activity in the myosin head. Activated myosin attaches to actin, initiating your power stroke. Release of your bound ADP is followed by binding of another ATP, and the absence of ATP prevents dissociation of cross bridges, causing rigor mortis. You'll see this figure on your right. For the contractile cycle, the binding of ATP to a cleft in the myosin head, detaching the head from the actin filament, and then the hydrolysis of ATP to phosphate group and ADP, which energizes the head. The head binds weakly to the actin filament. The release of phosphate group, which causes a tighter attachment of the myosin head to the thin filament, and the power stroke that moves the thin filament toward the center of the sarcomere. The release of the ADP, which sets the stage for another cycle. Now is the excitation and contraction coupling. The contact between nerve and muscle is called the neuromuscular junction. The linking of the nerve impulse to the shortening of the sarcomere is referred to as an excitation and contraction coupling. Action potential in muscles is propagated into the cell interior by what we call as your transverse or the T-tubules. The T-tubules terminate in proximity to the sarcoplasmic reticulum, which stores the calcium until needed. In a relaxed sarcomere, tropomyosin blocks the myosin binding sites on the actin molecules. To initiate the contraction of the sarcomere, the calcium binds to troponin, causing a conformational change. The movement of troponin is transmitted to the adjacent tropomyosin. This shift in position of the tropomyosin exposes the myosin binding sites on the adjacent actin molecules, allowing the myosin heads to attach to the thin filaments. Force or tension that the muscle generates depends on how much it is stretched. At rest, muscle can generate most of the tension. When stretched, the active tension drops. This length-tension relationship is important in muscle physiology as it impacts athletic activities and daily movements. Electromyography or the EMG measures action potentials within your muscle tissue, and what we call functional electrical stimulation or the FES will stimulate your muscle activity. So this is a graph showing you the length of resting length versus the tension or percent of maximum. So you have A, C, and B. The contraction of the excitation and contraction coupling. Patterns in living cells include various types of bundles as well as cross-linked and branched networks. The organization and behavior of actin filaments inside your cells are determined by the interaction of actin with a variety of actin-binding proteins. They can be divided into categories based on their presumed function in the cell. You'll see this figure on your right, and it is indicated in this slide. Nucleating, monomer sequestering, end blocking or capping, monomerizer polymerizing, actin filament depolymerizing, cross-linking, filament severing, and membrane binding. We move ahead with the cellular motility. Cell locomotion is required for many activities in higher vertebrates, including tissue and organ development, formation of blood vessels, development of axons, wound healing, protection against infection. Cell locomotion, as exhibited by the fibroblasts, share properties with other types of locomotion, for example, walking. As it moves, the fibroblast flattens itself close to the substratum and becomes fan-shaped with a broadened frontal end and a narrow tail. Fibroblast locomotion is seen by examining its leading edge, extended out from the cell as a broad, flattened, veil-like protrusion. We call this lamellopodium. As a lamellopodium is extended from the cell, it adheres to the underlying substratum at specific points, providing temporary anchor sites for the cell to pull itself forward. A stimulus is received at the cell surface. You'll see this figure on below, where it activates the ARP2/3 complex by another member of the wasp wave family. This ARP2/3 complex will serve as nucleating sites for new actin filaments. Once the filaments have formed, the ARP2-3 complexes attaches to the sides of the filaments. This bound ARP2-3 complexes will initiate side branching that extends outward. As a filaments polymerize, they push the plasma membrane outward, thus leading to the extension of the edge of your lamellipodium. Traction forces occur at sites where the cell grips the substrate and helps to pull the cell forward. The greatest traction forces are exerted last, just behind the cell's leading edge. Focal complexes that form near the leading edge of a model cell either disassemble as the cell moves forward or mature into larger, more contractile focal adhesions. With axonal outgrowth, the growth cone is highly motile region of the cell that explores its environment and elongates the axon. The lamellopodia and filopodia from the growth cone respond to the presence of these physical and chemical stimuli, causing the pathfinding axons to turn toward attractive factors and away from repulsive factors. Correct wiring of the nervous system depends on the ability of your embryonic growth cones to make the proper ring steering decisions that lead them to the target organ to innervate. In embryonic development, numerous cell activities are necessary for the development of the characteristic morphology of an organ, including programmed changes in cell shape, brought about largely by changes in the orientation of cytoskeletal elements within your cells. One of the best examples are the early stages of the development of your nervous system. Towards the end of gastrulation, cells of the neural plate elongate as the microtubules become oriented with their long axis parallel to that of the cell. Neural epithelial cells constrict at one end, causing them to become wedge-shaped, and the layer of cells curves inward. Curving is from contraction of a band of microfilaments that assemble in the cortical region just under the apical cell membrane. With bacterial cytoskeleton, there are numerous prokaryotes that are containing tubulin, actin, and intermediate filament-like proteins. PAR-M polymerizes into filaments that separate two sister plasmids, or the microtubules. CRES polymerizes proximal to the membrane and plays a key role in cell shape, next an intermediate filament, and FTSZ polymers form a contractile ring that helps separate the two daughter cells or actin, and that's being highlighted in this figure A, B, and C: PARM, CRES, and FTSC. And that's it. This is the Moving Bodies, a lecture about the cytoskeleton and cell motility. And thank you so much for watching this YouTube channel. Don't forget to like and subscribe and comment down your suggestions below. Have a nice day, everyone. Thank you.